Okay, hello everyone. Good morning, good afternoon, good evening, everyone. Welcome to this the first breakout session of HCA Asia meeting. So the theme of this breakout session is Atlas integration, technology, strategy, and the role of China. So I'm Xuegong Zhang from Tsinghua University, and here we have Dr. Ge Gao from Peking University. So we are the co-chairs of this session. We have quite a few panelists. So what this session will like, so we have some, we have proposed some topics for this session. It's more or less like a panelist session. So we invited a few panelists to give their opinions on the challenges and probably solutions, strategies around the theme. So I think I would like to start to invite all our panelists to give a introduction on yourself, like who are you, where are you from, and what do you work on? So probably I start from myself. So I'm Xuegong Zhang from Tsinghua University in Beijing. So I'm one of the co-organizers of this HCA Asia meeting this year. So I work on bioinformatics for human cell atlas and for other fields of biomedical applications, mostly from a machine learning point of view. So that was me. So I hand over to Ge. So Ge, you introduce yourself.
Hey, hello. This is Ge Gao from the Peking University. I'm a bioinformatician with focus on developing algorithm and tools to decipher the gene regulation, especially in the human and plants. And it's my pleasure to co-host this session with Dr. Zhang, and welcome.
Okay, now let's have our, so the order of this, I have a random order, so let's just get the name. Hae-Ock Lee, I'm not sure what the pronunciation is.
That's right, Hae-Ock.
Okay, Hae-Ock. Hi.
Oh, we go next.
Yeah.
Hi, I'm Hae-Ock Lee from the Catholic University of Korea. I'm a biologist working on mostly cancer, especially lung cancer. I use lung reference data, so I'm more on the user side of the references and atlases. And I like to put my inputs from somewhat different side from the bioinformatician.
Great, thanks. Now we have Jinmiao from Australia.
Hi, I'm Jane Yang. Yep,
Hi.
I'm Jane Yang from University of Sydney in Australia. I'm a statistical bioinformatician or data scientist that people call these days. And so I like to utilize the atlases, or hope to be. I'm more like a data consumer of the atlas, but I'm also hoping that as we develop novel methodology, we can contribute back to the atlas to enable people to use our tools to extract as much information they want from it.
Thanks. We have Wataru Fujibuchi from Japan.
Yes, thank you for introduction. My name is Wataru Fujibuchi from Kyoto University. I belong to Center for IPS Research and Application. So my main job is to contribute to IPS research enhancement. I'm a bioinformatician, so I'm developing a database for iPS cells and also human cells to enhance regenerative medicine or iPS cell-based disease curing. Okay, thank you.
Thank you. We have Jonathan Scolnick from Singapore.
Yeah, hi. Great, thanks. So I'm at the National University of Singapore. I'm a molecular biologist. And our group does a lot of assay development in single cell biology, adding to multi-omics tools that can be used. And then we apply those tools to understanding heterogeneity in tumors and how to then translate that into clinically changing the treatment of patients.
Thanks. So on my list, we still have a few other people, but they are not here yet. So if you are in the audience, like Long Qi from China, Shanlin from Australia, and Jinmiao from Singapore, if you are in the audience, please identify yourself with the host of this Zoom meeting room and get invited as a speaker. Or who I forgot to ask for introduction. If there is anyone I missed, please just turn on your video and talk. If no, I'd like to share one slide that shows the topics we would talk about. So does this work? Good. Are you seeing my slide?
Yes.
Yes.
Yeah. Okay.
Yes.
Thanks.
Looking good.
So as I said, so we will have about 1.5 hours to talk about Atlas integration technologies, strategy, and the role of Asia. So Ge and I listed some potential topics we may focus on, like major technological challenges, major technical challenges of Atlas integration, major strategical challenges for Atlas integration, and some of the existing or potential solutions to those challenges. I think the first three, I would like to ask Ge to host the discussion on the first three. And we want to talk about what should be the final HCA like. Or for example, whether it should be a centralized database or several distributed or parallel versions of the database at multiple centers, and if they are at either one center or multiple centers, who should fund those centers? And for the application of the Atlas, how could the integrated Atlas be used for medicine or biology-related industries, etc.? And especially because this is HCA Asia meeting, we are interested, we want to talk about the role that Asian scientists may play in this Atlas integration. And also we want to talk about possible legal and ethical issues. So those are the topics we want to talk about. So this is quite an open platform for everyone to speak on this. For the audience, if you have any question or any comment, please type in your question or comments in the chat room of this Zoom meeting. So that there is a chat function, you can just type in, we will see that. So Ge, I will stop sharing this screen, and so we just move on with these topics.
Okay, terrific. So Xuegong, could you keep the screen sharing on so we can read the topic?
Okay.
And, as a reminder, right?
Okay. Yeah. I will share it in this way because otherwise I will unable to see the chat room.
Okay. Yeah. I'm on mobile, so my connection is a little fragile, and I guess I can read the screen and everybody can. So let's start from the first one. That's the major technical challenges for actually as integration as I mean, in terms of the both computation and the bench work. So as to me, I believe the most observed challenge is batch correction because the batch effect is part of the we don't like the batch effect, but it's actually part of the reality. So we have to deal with it. And I believe we already got several amazing algorithms such as I say merge from the Dr. Yang, and I really like the paper and my students just present during last year's journal club. So could
Thank
you do some comments here?
Yeah, certainly. So thank you very much. That was a very kind remark. So our group actually in 2019 produced a work SC Merge where we do data integration. Now that was built on this concept of if you have like stably expressed genes and if you have the concept of pseudo replication, you could use factor analysis model to try to group data together. So if I can actually speak to technical challenge, I think there's still room. There's actually a lot of method out there. The real challenge here now is scalability, right? You can have a fancy method that looks at, maybe 10,000 cells, 20,000 cells, but with the Atlas, we want to go to millions. We want something that is not just good and stable and accurate. And there's always a trade-off between that and computation. With the Atlas, you want to go to million, 2 million, and you want to do it in reasonable time. And I think that's probably going to be some sort of innovation between computational statistics as well as, cloud computing type strategy. I also think that integration is not just in SC Merge, it's only one platform. It is only a single cell RNA-seq, and currently we're trying to generalize it to things like CyTOF and imaging with a lot of spatially resolved data in multi-omics. I think each of the multi-omics platform will have this technical challenge, as well as maybe we can borrow the information between different omics to actually help with this kind of batch correction. So I just want to say it out there. I think the technical challenge has blown up. Thank you.
Okay, so as to the scalability, you just mentioned, we actually have to deal with millions of cells simultaneously, or actually we can get a divide and conquer approach, such as this size by this size, or you think we still need to deal with all of those data, those massive data at the same time?
I think it's important to talk about the issues for the technical challenges and strategic challenges for the Atlas integration. A lot of times we think of the batch correction first, but I think it might be good to define what we want to do with the Atlas. When actually I got this topics, Atlas integration, I thought about like a textbook for the anatomy or histology. That's something like a stationary data. So we are trying to generate Atlas for different organs and tissues. And then there's another like a different strategy. It may not look like Atlas, but it could be like a reference, like a molecular signature database. So most of the times for the batch correction, we think about that stationary Atlas, the cell types, the definition, the annotation of cell types that don't change, not as dynamic as we think of transcriptome a lot. So I think it has a different nature. It's the same data, transcriptome, but for the Atlas, we want to generate some less dynamic data so we can use for different cell types, organs. And if you think about the anatomy or histology, they don't actually deal with the whole body. They deal with the whole body, but describing the whole body at one time, it doesn't really help to understand single organs or tissues, even for whole body. So I think it's nice to look at the whole cells from the entire bodies, but if you want to study it, I think it's more important to define like a tissues or organ levels more accurately than just trying to put in everything into one atlas. So I think it's more like how you want to generate the atlas question.
Mm-hmm. So the point is whether we would run the integration as Wikipedia or as GEO or Express, right? So in the first approach, we can collect the marker genes or signature genes, and which could be used to define the distinct cell types, and then we collect those information as a working collection, so that any newly generated data set could be feed in and run cell typing. So we can still get the cell composition and maybe the association between the cell type and the phenotypic variations such as tumor and non-tumor. And so Dr. Lee, is this your point? So just generate the atlas as a collection of signature genes to define the cell type, right?
Right, but what I'm trying to say, in a technical point of view, the batch correction there may be a different batch correction for like each type of atlas. If it's gonna be a whole body type of atlas, I think you may wanna use different batch correction than the organs or some like an immune cell type of data. So I thought that a lot of times we talk about the atlas generation, we jump into the batch correction. I think it's the right technical approach, but I think it may be good to think about what type of atlas you want to look at and what would be most helpful for the researchers and like a further studies in terms of using atlas. So I think the batch correction could be different depending on the layer of the data you are dealing with.
Great. So let me interrupt. I say Jinmiao Chen from Singapore joined. We missed you in the introduction. Can you introduce yourself? Turn on your video and introduce yourself.
Yes. Thanks for the introduction, Prof. Zhang. I'm Jinmiao. I'm from Singapore Immunology Network, and I'm doing single cell analysis, especially for human immunology.
Okay, thanks. Yeah, we will. I will share my screen again, and we are talking about the first three topics right now. I'd like to give my comments on that. I agree very much with Hae-Ock. Sorry for the pronunciation. So batch effect correction is an important issue, but it depends on the target of the atlas and the application. I think batch correction maybe ultimately it is not a feasible task. So because the technology involves always, and we have the history data, we cannot redo everything again and again. So I think the key challenge or key solution probably should be we should try to invent this kind of a batch invariant strategy for applications. So the application should be invariant to the batches. That will be the ultimate solution. Otherwise, at the data level, we can do it in small scale, but in large scale, it hardly imaginable when we have, say, technology in 5 years later, we want to do the correction with the data generated today. That's my.
So I guess it's really I really agree with Dr. Lee's view. It's it's in Australia there's a saying horses for courses. So what is it that we want? What's the question we want to ask? So the frameworks even I think the word batch correction is probably better used to rephrase it and say we want to remove, variation that we are not interested for the question of interest, right? So depending of your question, whether you're looking at disease progression or treatment, you want to remove that something that hinders you from looking at the signal that you're interested in. And also, I mean, there's a question on the Q&A that sort of asks, can we incorporate this kind of hierarchical organization of cells for integration? But that's a different kind of question too. This is saying that how can we identify cell type, which is a very popular question that people are asking. And yes, there are methods. I mean, a little bit of self-advertisement. We have a method called SC classified that actually takes into account of different layer of cell organization in terms of cell type identification. You could realize that some of us are interested, like Dr. Lee was saying, sometimes very well-defined cell type, but other times when there's perturbation, your interest could be even cell state or really finer details. And I would have to then leave it to molecular biologists who can comment more on those section. But can I actually ask Xuegong and Ji as the topic, what is the aim of many people have different views of what they want to use this atlas, right? So when we're talking about atlas integration, are we discussing the question of how people can utilize the atlas, or what kind of support we want to put on top of this atlas so it will benefit user?
Yeah, I actually think that's really the key question from listening to everything so far, is that there are many different ways to use the data, and everyone's going to have a different view. And so is the point of the atlas to just assemble all of these data sets in one place and provide tools that people can use to couple with their own data? Or is the point of the atlas to actually pull all these data points together and generate, say, a dictionary of cell types?
Yeah, great question, great input. I think probably we should, I'd like to share my view on what is the purpose of this breakout session before we go in deeper on the topics. I think we are kind of trying to collect the opinion of some leading participants in this field, at least from Asia Pacific region. Probably on this topic, these topics are, most of them are open, and maybe we can end up with some kind of opinion, kind of a white paper, or it's some publishable comments or perspective or things like that. So in the afternoon, I will give a report to the general meeting of what we talked about and what we agreed upon. And but if we are really good, we may collect some of our opinions to summarize into a publishable paper that can influence the society to share our view on the technologies, challenges, strategic challenges, and solutions.
Yeah, I believe there are already several, the serious challenges in the field, but I totally agree with Dr. Yang on her comments for, what's the purpose of the integration. Just like what Dr. Lee just said, we may have a multiple integrated atlas at the organs or tissues level instead of a holistic atlas for the whole body, for the whole human or the whole mice or anything else. But I believe such purpose would highly related and highly associated and with the biology aims of our interested biological questions. So Dr. Fujibuchi, could you have some comments? I know you are an expert as IPS, and your talk on the 3D reconstruction is also amazing.
Oh, thank you for listening to my previous talk. Yes, I have some comments from disease treating institute. Okay, as I work for IPS Cell Research Institute. Yeah, people are trying to cure diseases in terms of tissues or cells recently. So actually, one of my PhD students is trying to find the specific locations in human body where some particular disease occurred. Okay, so in that point, I think human cell atlas can contribute somehow, which they're the most important target cells, okay, for particular diseases. So when you combine other databases to Cell Atlas, that way I think we can utilize. I think we can make Cell Atlas more useful for medical doctors or pharmaceutical companies, or actually researching in some particular diseases. Mm.
We got some. Yeah. Okay. Yeah, we got some interesting questions from the audience in the chat board. So I will read one of them, both of them. So how can we incorporate the hierarchical organization of cells in the integration of cell atlas? I think Jane mentioned this. Right, this is one question. The other question is, hi, Professor, how to avoid overcorrection during data integration? How to accurately judge whether overcorrection occurs or not? Thank you. So that's it from Ming Dai. The first question is from Josie. Sorry, I cannot read the name. So anyone else want to answer those questions?
Yeah, I guess the Dr. Chen may be the best person here to answer the second question, because I just remember her nice benchmarking paper published early this year on the benchmarking of patch fraction, right? Hello, Dr. Chen, are you still there? Hello?
Or some other who want to talk about this overcorrection during data integration.
If I can jump in, it's hi. It's a really challenging question, and that's always in modeling. At what point do you stop, right? At what point have you, do you overcluster or do you overcorrect? So what happened when you overcorrect? When you overcorrect, then the signal just gone. Right, so signal is gone. So if you actually have, and so there's no one quick solution for it, but maybe one way that we typically do in our group is that if we have some sort of, if we're comparing, say, treatment and control, or we have some known positive, we make sure that the positive signal isn't removed, or the positive signal actually still stays there after the correction. And sometimes you can use that to tune the parameters associated. But Dr. Chen is here, and we should actually hear from her.
Yeah, Jinmiao, so Ming Dai in the audience asked how to avoid overcorrection during data integration. So we thought that you probably can comment on this question.
Yeah, so yeah, that's a very good question. I think Overcorrection to me, right? It means that the different cell types in different batch were overlaid together after the correction. So they're supposed to be a different cell types in a different batch, but after the correction, they are put into the same cluster. I think that would be the that would be the indication of overcorrection. So what we will do is we can annotate the cell types for each of the batches independently first, and then after the integration, and then we see whether these are cell types, the same cell types are clustered together while the different cell types still are well separated. I think that would be the way we would the methods we would take up to prevent the overcorrection.
So as to the overcorrection, you mean could we, identify the difference between the undercorrection and overcorrection, based on the cell types identified in the different this size before the batch correction, right?
Yeah.
So I mean, would they, would they imply some limitations that we have to I mean, we must have a comprehensive list of known cell types. So what if there is some novel type of cells in this size which we may miss? I mean, before the before our analysis.
Yeah, that is true. If there's any new cell types, that the task will be even more challenging. Yeah, so the methods we is using maybe is just only applicable to the known cell types.
Mm-hmm.
That's a very good question. Yes, yeah, I haven't thought into along that direction.
Right. We got new question from the audience. One question is about how about we have a lot of samples, and I think it will be labor intensive. And another question is could the panelists also comment on challenges in data projection reference mapping in addition to batch effect correction? I think this is a good question that is about the application of the atlas. So how we do the batch effect correction depends on how we do the mapping of new data on the atlas. So anyone, I suggest, time really flies. I suggest that after commenting on these questions, we move to the more strategic topics rather than the technical topics.
Yes, I agree. And so if I say the real question that I think the people are asking is how do we identify cell type? Look, a lot of people, I mean, when the field came about, they want to do a lot of merging because they can cluster and use the clustering, unsupervised clustering to identify cell type. But really, if you look at last year, and that's the power of this atlas, is if we are now having so much information in the atlas, we know a lot of information, we should utilize that information to train a model, and that way you can predict new cell type and map new cell type as much as you can, providing that you can sort out the scale. So there's a bit of challenging with the scaling issue, and as I said before, we have a package called SC Classify to actually take atlas data. We actually did take mouse atlas data as an illustration, put them together and use it to refine cell type prediction. We could also utilize, and I think this is the thing about atlas, a lot of times with atlas, we are surveying a huge number of tissues, a huge number of cell type. We may not have that refinement. Whereas there could be a data set out there somewhere where they're only focusing on a very specific lung and they have a lot of information and they manage to have a lot more rare cell type in it. I think as an atlas, we could also utilize those information to refine a notation in the atlas. I think that goes both ways.
Actually, I agree with Jinmiao that
we have to just make it clear the scale of the atlas, whether it's like a body level, cell level, organ level. So we can choose the level of integration we want to do. And I think like a technical issues, batch correction would be different depending on the level of the integration. If it's whole body, we may want to leave a lot of differences in like a tissue. So we may not want to use extensive batch correction for there, and we may not need this batch correction because the variability is relatively large for different clusters from different tissues. But if we look at the specific like organs or specific conditions, the batch becomes more issues. If we don't do the batch correction, we cannot really identify the real difference between the like groups. So I think it's really important to have a layered structure for this atlas. And we can use the specific layer for the specific questions.
Right. We got another question from Josie. So how could you account for inter-sample variation when integrating data sets? Can variation between individual effects integration performance and cell type identification from cell atlases? So that's the risk of batch correction, I think. So I'd like to share what in my lab, we kind of prefer trying not to do batch correction whenever possible, so that we can keep the individual signal. To just do all the pre-processing as carefully as we can. But when we do when the study involves multiple individuals, we try not to do the batch correction, and we find some other signal to indicate whether batch effect is serious in the data.
Yeah. So I know the multi-level of batch effect is a real challenge, and especially for the large data size, which may include the batch effects across different samples, across different individuals, and maybe across different technical platforms. So my lab just work on this by taking advantage of the deep learning, especially the adversary learning through autoencoder architecture. But it's still hard. So I really like the idea of the Dr. Lee that we may instead of a holistic one, we may have a multiple atlas which may be useful for the biological questions, but not too complex or too complicated to be handled by the current computational techniques. But I mean, I believe there is still other challenges other than the integrating the RNA-seq, the saRNA-seq, because they already have a multi-omics data, including the attack-seq, the mesosome, and maybe the proteome at the single cell level. So, I mean, is there any possibility that we can take advantage of those multiple omics data and use those data paired or unpaired to improve the integration? So I believe that's maybe the coming challenges. So what do you think, Xuegong?
I think probably we should move on to the strategic challenges. I think that it's relevant to your question that say at what level. I mean, it's not at the tissue or organ level. I mean, at what level? Say it's at the raw data level, or at the matrix level, or at the annotation level. So at what level we should do the atlas integration? So I would like to remind everyone that when we talk about atlas integration, it's not about a single cell study. It's about the human cell atlas. It's about the integration of data from multiple labs across many years. So it's not the data integration task in a single study. So probably we move on to the strategic challenges.
Right.
So currently, I know in the HCA data platform, they are collecting the raw reads. They are trying to do the integration starting from the raw reads. Do we think this is a good idea, or we should do at what level?
I think as raw as the data is possible, because analytical strategy and computational strategy will forever be improving, right? You do not want, and there's a lot of example database, TCGA, GO, SRA. The thing is, you want to be a good resources. You don't want to dictate what people have to do. Everyone will use the atlas in a different way, and they will have different method or different ways. There's just so many methods out there. We can't, as a resource, the best resource is if it provides flexibility and robustness and access. I think the strategic challenge for atlas to be useful in the integration is to standardize annotation. What information as people submit into the database? And I think Wataru probably have more experience in this, is what do we need as a minimum information going into this database so that it's not apple and oranges, right? I think as a consortium, as a group, this is where we can contribute.
So with Jinyang's comment, I think I'm slightly jumping to like the effort should be centralized or curation of different data set. I think it's all related. So for the atlas, for like in some way teaching view, it could be like the whole map type of things that will be centralized with the raw data provided. But for many like a biologist without bioinformatic background, the reference types, the molecular signature DB type would be very informative. So I think, again, it's the layered structure is my opinion on the atlas integration and everything. Like we could have like a whole atlas level and then like organ atlas levels. And it will be always good to have the raw data attached. But for the people without much of bioinformatic background, the ones that can be most utilized would be like a molecular signature DB type data. So they can refer to what they need. So that could be more curated effort from the human cell atlas. But for like the whole body or whole tissue atlas, the centralized data provider could be more appropriate. So
sorry if I could jump in. So kind of along those lines, I wonder if there's either a reason and a way to have something similar to what Galaxy has done for sort of standard genome and RNA sequencing for people, as Dr. Lee mentioned, that people who don't have bioinformatics background, but could still then utilize these data sets if there are some standardized workflows that at least can be built in so that people can take data sets that are in there, plus maybe some data sets that they also develop.
I thought HCA has a data platform group. No?
Right. It has a data platform group, but so it's that's related with the fourth topic, that is whether the HCA will be a centralized single database. I know there are a lot of single cell studies that have not been deposited into the HCA data protocol. I don't think currently there is a mechanism for everyone to contribute data to that. And there are quite a few efforts around the world trying to do data integration by themselves. Like in Ge's lab, I think he did a database on that. And I know some others, of course, in my own lab, we are also doing something like that. So there probably we jump to this multiple center thing. It's kind of unavoidable. We have multiple centers. They may not be necessarily officially called HCA, but it's something like HCA. So what is it? So it's unlike, so people like to compare HCA with Human Genome Project, but Human Genome Project is very centralized effort, and the samples are from the same pool of 3 individuals, and everyone in the world are sequencing the same sample, part of the same sample, and their data integrated into NCBI. But now people are working on different individuals, and even in the same lab, it involves multiple individuals, donors of the samples, and then this then probably it's we should have some discussion on what will the final HCA look like in our mind.
I remember I was at the HCA Asia meeting last year in Singapore, and I think a similar comment actually came up, is how do we get people to submit or actually contribute the data into HCA, right? So
Right.
I'm interested in hearing, most of the time, all data will be generated. So I'm going to say, like, I'm not part of this in the sense that I'm a data consumer as much as I'm not a data contributor, because I don't generate my own data. But I understand that in most countries, the government funds, or there are some funding to fund the data generation. So if you, for example, are funded by CZI, I think then you would be putting this data into the Human Cell Atlas. Is there any advocacy group that one can form to talk to the national funding bodies that mandate or strongly suggest? I think they are doing that in Europe, because if you can actually mandate the national funding body is like, yes, if you want money from us, you have to contribute to certain, whatever they ask you to do it, you will do it. Right. That's how I think Geo got populated, right? If someone say, you got money from us, you have to put it in this database, then that data would just get in there. Or if journals, that's the other way. If journals are willing, then the data will happen. So it's either the funding body or the journal. So I'm not sure, maybe Xuegong, you guys could comment a little bit more. Is there certain subcommittee that can be formed as an advocacy group in the different country that talks to the funding body?
Most of the funds in Korea comes from the government, and many of the genomic study, the government actually mandate the data deposition, but that's actually within Korea. So if it's gonna be publicly available worldwide, I think there needs to be more like a worldwide advocacy group. And I thought that a lot of times HCA wait for the data contributor. Is there a way that from the HCA contact those with large data set and ask for the data deposition or contribution? I don't think there's the HCA people here, but
Yeah.
I thought maybe for a large well curated data set. So maybe there could be a time that HCA collect those type of data and provide a curated data set.
So just like a meta database, which integrate multiple distinct databases curated by a uniform standard and provide a unified interface to the community. Do you mean that? I believe.
Well, at certain level, when we submit paper, most of the times we are mandated to deposit at least the processed data, and many times raw data at controlled access. So data are there. For like a patient data, it's difficult to have like a purely open situation, but for the reference types, it may be more durable because of the de-identification of all the information. Actually, my topic has to go to like an Asian specific part again, but from many Asian countries, there's not as much reference type data contribution actually because of difficulties of getting it. So the most of the data from Asian countries are in controlled access because most of them are from the patients. So I think it somewhat prohibits like the complete release of the data set.
Right, that is relevant to the strategic or the system of rewarding for data sharing. I think nowadays, so many labs really are eager on the credits they will earn if they contribute the data. And the current system is that it's not well, rewarded if some lab contributes data. Then that's the difficulty. And also for the funding agency, that's why people are really interested in publishing their data in papers, but not really publishing the data, releasing the data in common databases. So they try to meet the minimum requirement of the journal to submit the data, but the data is not really at the level HCA expects. Then that I think is a kind of a centralized human cell atlas, just like the Human Genome Project. It's not that feasible. I mean, Human Genome Project was quite successful, but HCA, it is hard to do it in a similar way as a Human Genome Project. That's my observation. I know a lot of people, so huge amount of single cell data are outside the HCA platform.
Hello. Yeah. I believe there are many other concerns, just as what just raised by Dr. Lee on the privacy. So, given the fact that many data were generated based on patients' data, so like the cancer, diabetes, or a lot of diseases. So I believe there may be some, concerns on privacy issue, and especially without the, clear regulation on the ownership of those data and on the permission of usage of those data. So I think that may be especially challenging for human atlas because we are dealing with human instead of mice, right? So as to Hello, hello.
Yes.
Yeah.
Yeah.
Hello.
Go
So
ahead.
I would add on this that is I think the reality is that when we talk about Atlas integration, we must really analyze the reality of data generated. Most of the single cell data generated nowadays are kind of the product of some scientific project on a specific scientific question rather than building the atlas. I mean, I have said this in Singapore last year. So building the atlas is not just doing a lot of single cell scientific research. It's a different, it's quite highly relevant, but it's not equal. So it's different from say, tens of thousands of single cell papers will equal to a human cell atlas. So that's the challenge. So each piece of the data are the product of a specific scientific project that may not necessarily aim at building the atlas. Now we need to integrate the data of that nature and try to build the atlas. I think that's my consideration for this. And it's challenging. It's a challenging for the technology, but we really need to face that. It's not an ideal design as the Human Genome Project had.
So as to the medical application, such as was Dr. Lee just mentioned, such as on the cancer study. So how those data were generated and used in real, the cancer-oriented studies. So I'm a pure bioinformatician, so I know very little on this. So definitely, can you comment on this?
For our data set, actually it was mandatory to deposit in the government. So it was released to Korean researchers, but without much curation, without much annotation. But then when we publish the paper, we have to deposit the data that public can use. So we deposited the process data to GEO, and then the raw data as a controlled access because it contained the whole exome sequencing as well for the patients. We deposited in the European Genome Phenome Archive. So people are asking for the permission. They have to write the data transfer agreement with the institution the patient visited. So it's to track and to, let's say it's a cancer data, but let's assume that we integrate into the HCA data. So I think I may need to write like a data transfer agreement with the institute and ask for the permission to, like, again, deposit the data to the HCA for the public open, for like a entirely open policy if we are gonna follow.
So it's a requirement of the government or the funding agency.
So so it's government funding agency, the Ministry of Education and Technology. So as far as the Korean law, we had the institutional review board for the data generation. So it's okay to use the data, and the government actually asked us to deposit the data for the public use. So that's fine. I mean, for the opening the data to the public. But
so how about the regulation in Japan? I mean, Dr. Fujibuchi, could you comment on this?
Well, I'm also a bioinformatician. I'm not very much familiar, but I used to work for GEO. So, so when you publish some data from experimental researchers, then you need to submit your data as well to those public places. So for example, GEO or EBI, those databases. But this has a very complicated issue unless you give a license, because whenever you make some data, you are owner of this data. So you are given a license for this data. So unless you give your data to HCA and any platform, nobody can utilize your data. Okay, regardless of it's a patient or normal or non-human data.
So Jen, you said you would leave earlier. So do you have any comments on the topics we haven't covered yet? Unmute. Yeah. Yeah, you're muted.
Sorry, I just no. Apologies. I was trying very hard to stay, but sorry, I have to go. No, I don't have much to say. I think it's actually really a big challenge, and hopefully, I'm looking forward to Xuegong your report about the group discussion, the combined report from our discussion, and hopefully, I think there is a real benefit if we can get some things going. So I'm interested in, what is the product eventually we'll see from the HCA Asia.
Okay, great.
Thank you.
Thank you.
So probably let's move on to the other topic. We already covered some of the topic 5, and we continue to talk on that. And now probably we focus more on the role of Asia Pacific region can play for the human cell atlas. And we know currently everything is dominated by the US and European scientists. We are just kind of following and contributing a little bit. So let's move on to the remaining topics.
So as Xuegong mentioned that the reference type of data, there is a very few contribution from Asian countries. One of the reason is the difficulty of getting material for the reference type of data. So even the funding from the government focuses on the disease, not really this reference type of data. So we have to find funding for the reference data. We have to find the resources. And actually recently in Korea, I found a way to use the de-identified material from the forensic medicine. So maybe we could start generating the reference type of data and contribute for this human cell atlas type data. So personally, I was interested in like a normal reference because we have to compare it to the disease state cancer, and we wanted to use reference that is generated in the same process as we generate our cancer data. That's why I pursued the reference type of data generated by our group. So in this way, we can contribute the reference data to the HCA. I think there may be other researchers in Japan and China that has similar interest and try to pursue generating reference type of data.
Jinmiao, do you have any input about the situation in Singapore?
Hi, Prof. Zhang. May I ask you to repeat what's the question?
So that's open. It's about the kind of the role of Asian scientists that can play in the atlas integration and whatever other topics.
Oh, so in Singapore, I think the Human Cell Atlas is led by Shyam. Shyam, he has this flagship projects on the Asian human cell atlas on the immune cells in the blood. So I think yeah, I'm not part of that, but I think Shyam is the leader in Singapore for the human cell atlas Asia.
Well, what about the general policy from the government funding agency? So in Singapore, most of the scientific fundings are from the government, right?
Yes, most of the fundings are from the government, and we try to apply for government fundings for the Human Cell Atlas project, but not successful because the Singapore government would like to see more translational research, meaning that a research that can be applied to the clinic or to the drug or to the company. So to Singapore government, the Human Cell Atlas project is very fundamental. It's very fundamental, so we don't see a direct revenue or like translational application from this project. That's why it's very, very difficult to get our fundings from Singapore government for Human Cell Atlas project. At least based on our experience in the past applications, we submit multiple times but rejected. we were not successful.
Yeah, that was a similar situation. Lee mentioned in Korea, and I think it's similar in China. So, so that's why I said that probably from the bioinformatics aspect, we should try to invent technologies or methods to integrate data that were designed for disease study or some other developmental study rather than reference purpose. But we try to convert those kind of data to some reference. That's challenging, but probably we need to face that reality.
Yes, Prof. Zhang, that is a very, very good point. That's a very, very good point. In terms of bioinformatics or data analytics, Singapore government is very, very supportive. They are throwing a lot of money on AI. So I think we can get the grant if we come up with a project to propose AI methods to integrate a Human Cell Atlas project. I think we have a chance in terms of bioinformatics and dry lab analysis. In terms of that aspect, Singapore government is very supportive.
And probably Shyam's application for that, the immune project could be a good model, so we can form our international consortia and apply for the CCI grant for the reference type of data for other tissues. Then we have to have resources for the tissues, but I think there's a way to get funding. So we may try harder to get make the international consortia to apply for the grant.
Yes, yes.
But following on to what Jinmiao just said is we can, as a group, if you are talking to individual labs who are doing specific projects. So for example, there might be a lab doing a project where they're looking at liver cancer. So that's kind of popular in Singapore, but obviously they need normal liver or adjacent normal tissue to be able to do their work as well. And that's sort of the underhanded way to be able to fund exactly that type of work. So it may not work to say, we think the government should fund the collection of all this normal tissue for the purpose of making it publicly available. But if HCA members just as a group can sort of have, the handshake agreement that says, well, every time I do my cancer study or whatever study about a particular type of tissue, I'm going to take my normal tissue and be able to provide that data to the HCA. You can kind of get to the same place.
Yeah, as to the data integration from the public data, I believe our bioinformaticians could do a lot of things, such as not only the batch correction, but also the data curation, because just like Xuegong just cited, thousands of millions of billions of the single cell data are just being generated every day across the world. So we actually can take advantage of those great sources as an input to our algorithm, such as my lab just work on atlas named HCA, or Earth Cell Atlas, which aims to integrate multiple sources, including not only the human, but also the mice and the fly, so that we can have a unified view with not only the cell data, but also the cell type curation based on the cell ontology. So I believe, it's sort of challenging, but just as Xuegong said, given the realities that not many funding agencies or the governments would give immediately support on such basic studies in the Asian countries. We bioinformaticians can still work on some public data and also present those data as a integrated portal for the community. But I believe that as a long-term goal, we may also seek to get more support from not only the local governments, but also from the funding agency internationally, like through a lot of collaboration projects between different countries. But I know it's hard, but I think we can take advantage of unique resources in the Asia on the diseases and healthy sample, and just like what we just discussed.
Yeah, I saw one comment in the chat board that from Katie Borna. I agree, for a human reference atlas, we need to agree on the minimal metadata that should be provided for each tissue or data sets. The metadata might differ by organ. For example, smoking and other environmental factors might only affect certain organs. So I think that raises an important technical point in the strategic study, that is that we need to kind of define a minimal metadata so that it can be the data can be integrated. So now let's move on. Is there any consideration on legal and ethical issues?
Well, as far as we are concerned in Korea, it applies the same rule as for many other genomic data. So there are still ongoing debate how we are gonna make it publicly available, the genomic data. So it's in the same line. So there are still issues like limiting the metadata, like a patient data or individual data available to the public. It's very limited. But again, with the controlled access, many metadata can be deposited and make available to the researchers, not really open to the public. So it's a same rule for the other genomic data? Probably same for like China or Japan, or is
Right.
there a specific rule?
Yeah, in China, kind of the legal environment is getting more stringent on sharing genomic related data. We need to do more paperwork when we want to publish the data. So basically.
And in Japan, the same thing for genomic data. But for transcriptome data, it's a different, more free.
But sometimes it's hard to define. So because the lawmakers, they are not scientists, they don't know much about the difference between the transcriptomic or genomic. So I don't know the situation in other countries, but in China, so it's a kind of as long as you are talking about anything about DNA, RNA, and they think it's a kind of a genomic data. It's that's why we need a lot of paperwork to explain that.
Is there a way to, in some sense, kind of draw a red line to say like the raw sequencing data, because that, even though it's RNA sequencing, right, obviously it contains genomic data, and maybe that falls under sort of one set of regulations. But if you had in, say, RNA sequencing, if you were just to look at the matrix tables of expression,
Right.
then that doesn't really lead you back to, couldn't really lead you back to an individual. So is there some way to have that kind of strict line? I mean, ultimately, as was discussed earlier, we want all the information possible, but as governments are sort of becoming more strict about the genomic sequence data, is there still a way that we can get very useful information out without that regulation?
I think sharing metrics, I mean, the expression level data is easier than the raw reads.
Yeah, because what I see right now a lot of times is that people are sharing the raw reads in places like Geo, but then they might get fenced in to where you can't access it. And for whatever reason, they didn't publish just the expression data because they were, in a sense, trying to be more open, but then that caused a problem. So I wonder if even just like a position statement from the HCA that says these multiple data types should be deposited into databases so that we don't have this problem of data getting locked up might be useful.
So we have 10 minutes before finishing this session. So I think I open all these questions to everyone. And if you have some further comment on any of the topics, you are welcome to share in the last few minutes.
Well, I think there is a very important question residing in this big project, which is Dr. Yang mentioned in this session. We don't have any method to define cell types. So we don't have a definition of cell types so far. So we have only biological knowledge from history. So, but we have a bunch of data here. So how can we correspond our knowledge to, to newly coming out big data, and how to decide which is which, how to define these cells are, that cells we already know. So I think this is a kind of a chicken egg problem, since we don't have a definition of cell types, so we cannot define, we cannot identify cells. So how can we solve this very important question? So how can we generalize the method to identify cell types? That is my big question in the data integration. Do you have any idea? Since we have many new cell types, which is discovered in the process of sequencing, but we don't know. We just, how to say, not objectively, but we subjectively define cell types so far. But is this correct? Is this correct, the cell subtypes?
Yeah,
I have a question on that.
I agree very much on this question, although I don't have an answer. I think at the current time, it is very important to read these questions and clarify these questions, identify these challenges. I think this is so it's equally or even more important than answering them, because many of the current publications, we see that they try to answer questions that have not been really well defined. So that's the thing that we should get our voice heard. Any other input or comments? For the audience, if you still have some other question or comments, this is a final chance for you to type in your comments in the chat bar. So if not, probably I should kind of conclude. I will try to summarize what we have talked about to see whether we can get an outline of this breakout session, the conclusions or kind of of this breakout session. And as I said, I probably will email you after this meeting. If you are interested, we can try to collect input from you, from every one of you, and we try to sum up what we have talked about and our thinking and our proposals or our questions on that. Of course, it's totally free to everyone. If you are not interested, you just ignore my email. Otherwise, I would probably would ask you to write down something for the topics we covered, and then let's see. We may not succeed, but we let's see whether we can write up something in a few weeks or a couple of months after this meeting on these topics, and give a kind of our opinion, our view on these topics, and to share it with some kind of audience in some area. So any other input? Ge, you have anything to say?
Yeah, I'd like to thank everyone for your great comments and great insights. And I wish we can get something new and more insightful as part of the white paper or even a journal perspective in the, I mean, just like the Xuegong said, in the next weeks or couple of months. Thank you.
Thank you.
So so thank everyone, and this is quite late for some of you for your lunch, and we don't have more questions. So let's just conclude now, and I will see you back in the plenary room this afternoon.
Okay.
Yeah, thank you.
Bye-bye.
Bye-bye.
Bye.
Bye.